Organization is key to the success of any program, and Mercer on Mission Ecuador is no exception. An organized notebook is required by all students to ensure a proper record of all of the fieldwork done. When the fieldwork is properly recorded in a student's notebook, a collected sample is issued a notebook number that corresponds with the date and time of the sample being taken and location of the sample, which is necessary to provide meaning to collected data. In addition to noting the location and date on every page, having every entry within the notebook labeled with the author's initials, page number, notebook page, and entry number per page provides for easily referenced material. This way, every student is allowed access to the details of every sample taken over the course of the research. The contents of every notebook that is used are typed into a Word document each day to create a second copy in case of an accident or a loss of a notebook. Having this typed report of the notebooks creates a safety net of information on procedures used in the field and data points such as water and soil samples. An important goal of Mercer on Mission Ecuador is to determine the amount of contamination from processing plants within an area's ecosystem. This goal is accomplished through the collection and analysis of air, water, and soil samples. Many different samples were taken from various locations in order to determine the effect of contamination from metals like lead, arsenic, cadmium, and mercury in the area's rivers and soil. A variety of locations will provide us with a preliminary understanding of the spread of contamination from the original source. From this data, it may be possible to determine how an individual processing plant's lack of waste management is affecting the local environment and population. The water and soil samples will undergo chemical analysis via ICP-OES and ICP mass spec to determine the concentrations of heavy metals in the environment. The information provided through these analyses will allow us to determine human and environmental exposure to these toxins. In addition, we cultured bacteria from the soil and water to determine if bacteria are present that are capable of methylating mercury. In the ecosystem, methylmercury can bioaccumulate in the food chain, which can be dangerous to humans. The bacterial cultures are also tested for resistance to mercury. Such traits can be related to antibiotic resistance in environmental and human pathogens. This provides an additional concern with excess mercury found in these communities because pathogens may become harder to treat.